Welcome to Biology 159 and the first video in our video series. In today's video, I'll introduce you to the basics of science investigation. In today's video, I'll introduce you to the basics of scientific investigation and experimental design. Today, I'll discuss the difference between a null hypothesis, a prediction, and a scientific theory. I'll describe why replicates are needed in a biological investigation. Summarize the difference between process and sample variation. Distinguish between a mean and standard error of a dataset. Explain how two means are determined to be statistically different. The scientific method begins with observations. You observe a system that you're interested in. You then gather background information about that system or that observation. You identify questions or problems that have not been addressed in that background information. And then you form hypotheses about these questions. A hypothesis is a proposed explanation for a phenomenon being studied. Scientific hypotheses are testable and falsifiable proposed explanations to account for observations made. Scientific hypotheses are testable and falsifiable proposed explanations to account for observed patterns or trends. Falsifiability means the capacity for a hypothesis or theory to be shown wrong. Falsifiability means the capacity of a hypothesis to be wrong. This prevents us from drawing conclusions in the absence of evidence. The null hypothesis is a hypothesis that there is no significant difference between The null hypothesis is the hypothesis that there is no statistical difference. The null hypothesis is the hypothesis that there is no The null hypothesis is the hypothesis that there is no The null hypothesis is the hypothesis that there is no significant statistical difference between the specified population or observation. After we have our hypothesis and we know what our null hypothesis is, we make a prediction. Predictions are statements about patterns, trends, or differences between groups. Ideally, for any hypothesis-driven question, you will want to generate multiple hypotheses, which you can then attempt to falsify by generating predictions, collecting data, and performing statistical analysis to determine if your predictions are true. After you've made a prediction, you then test your hypothesis through experimentation or observation. You then interpret the data and you communicate the results. Whether or not your hypothesis is true, it does not matter. You still communicate what your results are. Now, another point of confusion for a lot of students is what exactly is a theory? The common everyday use of theory is kind of like an idea or a hunch. So like somebody says, well, my theory is that it will rain today. That's not what we mean when we talk about a scientific theory. A scientific theory is a coherent explanation for a phenomenon in the natural world that has been repeatedly verified through experimentation, and it's used to make predictions. This is different from a law, which is predictable, observable phenomenon that a theory explains. In science, we have two sources of variability. You have process variation, which is natural variation among organisms, meaning between and among organisms. So you have intraspecific variation, which is differences between individuals of the same group, and interspecific variation, which is differences between individuals of different groups. So an example of an intraspecific variation would be differences between someone with a population that has mostly people with brown eyes and a population that has mostly people with blue eyes. You would be looking at both populations would made up of human beings, but you'd be looking at the differences in traits. An interspecific variation would be looking at like the differences between humans and chimpanzees. These are two different species, so it's differences between two separate groups. You also want to make sure you take as many replicates as possible to capture this variation. Sample variation is a measurement error. Be as accurate and as precise as possible to avoid these types of variations. To capture the amount of variation in data, scientists like to calculate things such as means, median, and mode. For our purposes, we will calculate the mean, which is the average, by adding all replicates you sample and dividing by the sum of your replicates. So if you take 10 observations, you want to add those 10 observations up and divide by the sum. Take that last part out. They also take measures of dispersion, so standard deviation. And this is the statistical measurement of dispersion of your data around the mean. You also take the standard error, takes the sample size and standard deviation into account. You also take the standard error, which takes the sample size and standard deviation into account and can therefore be used to quantify uncertainty in the mean and is defined as an estimate of how closely the sample mean represents the true population mean. We're going to stop here.